going to speak on. You can see his introduction to the proof of genetic engineering, which is a topic of some great comments.
Bill? Yeah, things have changed drastically for me. And in that April of so I was
before Melanie died, then?
yes,
Oh, I didn't know that.
yes, and I was recovering for a couple of months, and I've still got rather a big scar right down here. But thankfully, it restored a lot of function because I was at the point where I was my quality of life was going down. So thank goodness I recovered from that, and then of course my situation changed drastically last year. Rattling around in the house that formerly had 4 people, there's just now me, and the cat's even gone.
Oh. Hey, I got some.
Oh, it's okay. I mean, I used to talk to the cat, but you know, our conversation. But it's a pleasure to come back and talk to you. This actually, I prepared this presentation for the Azalea chapter of which is the local society. And you've had a couple of the members of that society come and give you talks already. So anyway, we'll see. Just so that you know, I'm not from here, in case anybody thought I was. I'm right here, Kingston upon Hull, in the county of Yorkshire, on the east side of the country. And it really is.
No, America's a part of the
Well, I mean, okay, well, I'm not going to get involved with that. I was educated at the university and at the University of Minnesota. And it was 1976 when my wife and I and 18-month-old son arrived on the shores of this country. And I worked for a couple of years as an assistant gardener at this park, which is a 15th century hall. And they had a herd of deer running around in that park also. So that was my first chance to consume venison when I got stuck on a fence and had to be destroyed, as they say. Killed was the word that should have been used, because they destroyed, because we took it in the employees took it in the little shack that they had and divvied the stuff up. No, having left the University of Minnesota, I went to work as a senior scientist in a plant biotech company in Utah, and my job was to get projects to think projects up and get them funded. Basically, they weren't interested in publishing anything. They were just interested in finding out what needed to be known to get to products. So I worked there. I managed a research tissue culture lab and developed methods for reproducing plants through that had a project with tissue culture. If anybody's never done any tissue culture, you generally use jars that fit in your hand. Tissue jars, jars that would eliminate some of the and some of it make the handling easier. The ability to culture plant tissues and organs in 5-gallon water jars. For tissue, largest one we had was like 50-gallon drum that we put Alstroemeria propagation material in there, and we ended up filling that thing with the Alstroemeria plates. It's never been applied on a large scale. The company I was working for ran out of money before they were able to working on. So we had a regular commercial tissue culture lab. Cost maybe a couple of $100,000 to build one of those, maybe even more nowadays. And of course, it's having the tissue cultures that you use for multiplication. If they're already contaminated, they've already got a little bit of bacteria in, well, then you spread the bacteria. So one of the most important jobs is to inspect them. And that's the job. You have to look at first signs of bacterial contamination. We were able to produce plants out of tissue culture, which then we were able to establish using page over a covered flat. It doesn't have to be the comics page. Could be the sports pages. It's just reducing the light, because when a tissue culture, it's like a I guess it's akin to a newborn infant environment to nurse them through. And this would be a more high-tech approach to increasing the humidity around tissue culture plants using a high-pressure mist system. Produces droplets of water similar to a cloud. So it's like walking out of fog when you're going to a greenhouse like this. So that's my background, where I came from. And then, of course, I went, after working for that biotech company. Yes. Different? How is it different to? Oh, well, plant tissue culture takes place in a sterile environment. So you have, let's go back over here. See if we can go back. Yeah, this bench here is a stainless steel bench, and this screen here, there's air moving in a laminar flow way bench, and that air is filtered to exclude the microbes. So it's sterile air. There's a stream of sterile air flowing this way, and everything that's inside this laminar flow bench should be sterile. Of course, the person's hands, when you put those in, they're not sterile. So normally you have to wear gloves. It's a high-tech method. the tissue culture, you need a defined medium containing nutrients, gelling agents, sugars, vitamins, those kinds of things on a defined medium, which is contained in test tubes or some kind of vessel. And all the work is under this sterile environment. And the tubes are incubated in growth rooms that are usually lit by fluorescent lights and air conditioned. So, I mean, you can sense a rooted cutting you can get for 5-10 cents. Plants start buying those, even if it's a foliage plant, they're going to be 70 cents. So it's 7 times more, at least 7 times more expensive technique for producing plants than regular propagation. And regular propagation, you're not in a sterile atmosphere. I mean, you want to be clean. It's impossible to be sterile in a greenhouse. Yes?
So it's basically for breeders?
Which?
The sterile environment ones?
No, they're not.
Well, then what's the purpose?
Some of them are used to propagate otherwise. That maybe you can only use division. So if you take a plant divided, maybe you get 4, 5, 3 or 4 months or something. With tissue culture, you can get a lot in a relative. So it's a production technology as well as a research technology. If you like, it's one of the important underlying technologies that enable genetic engineering to actually take place. So there was several technologies. This one allowed cells to be cultured reliably under sterile conditions.
Question now, is this anything to do with the space program? Are they going to try to propagate plants and maybe we need like that? No.
You know, I don't know.
Oh.
That's the simple answer to that question. I would imagine that the space people. Oh, that's the wrong computer. I wonder why it did work. Talk about plant breeding. Plant breeding. So, it's you use natural process. Plant is transferred by some method to the surface of the stigma. The pollen travels down the style to the ovules, an embryo, seed, etc. Those technologies have been around for centuries. Plant breeding is a big pro. I mean, corn yields went from certainly increased annually, while public plant breeders were working. Nowadays, you can add things like mutation breeding, and you take a part of a plant subjected to forces, if you like, and then select out any new plants. That people do that with chrysanthemums, carnations, roses, alströmerias, vegetatively propagated plants. Polyploidy, that's increasing the number of chromosomes by adding complete sets. Could be 1 set, 2 sets, 3 sets. Daylilies is a prime example of that. Using polyploidy.
Well, we haven't found that it exists on all the show.
You don't use it. Rescuing embryos from incompatible crosses and just plain cloning. This is, I put this in because I like this picture. One of my favorites. And besides, it shows you if there's any doubt as to what, where the sex organs are on a plant. This flower here will, as an example, because these little protrusions are the stigmas. So this is a fruit, and there's the stigmatic surfaces inside what is now the receptacle, which will become the fruit. And these things right here are the stamens that produce the pollen. So this is the, this is the classic picture of a hermaphrodite flower. A lot of flowers are like possessing both male and female organs, so it's therefore hermaphrodite. All right. And genetic engineering, there's a definition here. Genetic engineering is a technique using material of living cells. And a gene, in case you've forgotten, is a segment of DNA that expresses a particular trait, and it's a unit of hereditary. Moves from generation to generation. So once a plant is transformed, the seeds contain that evidence of the same gene, and it can be expressed in the progeny. Okay. Genetic transformation methods are built upon advances in areas of endeavor. Genetics, the science itself, that's some of the tissue culture methods, recombinant DNA technology, and so there was a public put into the research in the 70s and 80s. And I was, when I was at the, you couldn't write a grant for a conventional breeding program in the 70s, in the mid-70s, mid to late 70s. The funding agencies, technological. So they spent millions of dollars in public funding to transform plants. So, what do we mean by genetic transformation? It's using specific genes isolated from living organisms that are compatible with plants. So you can't move genes by quote-unquote normal sexual processes. So you have to figure out a different way to do it. And it's usually that does with the desired plant. Okay? And genes can be transferred from any other organism. You can have genes from fish expressed in a plant. You can have genes from viruses expressed in a plant. Genes from humans maybe expressed in a plant. Oh, that's not been reported yet, but there's no reason why you couldn't do that other than ethical and moral reasons. Technologies are there already. You don't just find a gene that says, oh, this is, you know, this will enhance the protein production in beans by, you know, 25%. Here's the little gene, put it in the bean cell. The genes come packaged. They come packaged with other enable the process to work. Because so most of the time you're trying to insert a gene into a single cell. And then you want that single cell to start multiplying so that you could separate that cell from the ones that don't contain the gene that you want. So if you've got, say you've got a cup of medium with cells suspended in it, you know, billions of cells. A small portion of those will be transformed in a transformation event. So then you have to select out the ones that are transformed, done by killing the ones that are not. And usually the gene of interest is a gene for antibiotic resistance. After you've done the transformation event, you transfer to media containing antibiotics, which kills the cells that are not transformed. It's pretty simple. Kill off the cells that are not transformed, and then you take the cells that are transformed, culture those up, and then from those cells, from clumps of cells, you want to form something that will give you a whole plant. So you can form a shoot, one that cells relating together and becoming a meristem, or you can get cells to form synthetic embryos. In other words, they just, for one reason or another, decide to go ahead and behave like they're an embryo. Not all plants are going to be able to do this, but many plants can. You just have to find out how to do it. And it's not easy to find out how to do it. It's laborious. But in the end, you will, if you're diligent, you will find media and techniques that work. So, and it's some of these other genetic resistance, gene from a bacteria or from a virus. It's some of those other genes that people are worried about. It's not just the gene that says, we'll say Roundup resistance. There's a gene that breaks down Roundup, produces products that break down Roundup inside the plant. That gives them. But it's also a package with these other genes from viruses, and so what do those genes ultimately? Maybe nothing. Maybe it's just protein. So it's typically transferred either as a circular piece of DNA, just DNA itself is coated onto a particle of gold, and a particle of gold is shot through a culture of cells. Particles of gold containing the DNA, some of those will hit cells and remain, and that DNA then becomes incorporated into the nucleus. We don't know how, we don't even know why, but it happens. So if it works, stick with it. So the point is, you can transfer genes from one taxonomic unit distantly, bacteria. You can do that with using these man-made, more than one gene becomes incorporated. We tend to talk about one trait on these pieces of DNA that get incorporated into the new recipient cell. To another organism. Not in the living world. You've got some of the nastiest organisms that we can find. Viruses, for instance. Viruses infect cells by putting a piece of their own DNA into the nucleus of a cell. It immediately takes over the cell functions, and instead of what they're supposed to be, the cell then devotes itself to reproducing virus particles, which kill them, and then the wall dissolves or falls apart, and the virus particles are released into the organism. So viruses already have a way of moving their DNA about. So do some bacteria. Plant diseases. The plant is tumors. Tumors on stems of roses. Tumors, swelling. I'm probably speaking English. You would think after 40 years I would have learned the language. Bacteria makes tumors on plants is a ubiquitous organism. It exists in the soil. It squirts. This is the agrobacterium cell, right? This is its DNA that does most of the work for everything that's inside the cell, except it's got this circular strand of DNA here. And when this cell approaches it has the capacity to dissolve a little hole in the cell wall, take this plasmid and squirt it, or take this little bit of the plasmid, this little bit removed, and put into the host cell, and that becomes incorporated into the nucleus, the nuclear DNA, and then the genes on this are expressed. When the genes on this plasmid are expressed, it reproduces itself to produce callus, big, massive cells. Mm-hmm. And that's it's called two agrobacteria. This one, Agrobacterium tumefaciens, and then this Agrobacterium rhizogenes. And what do you think that one does? Rhizogenes, does it give Diogenes Club? Nothing to do with Sherlock Holmes. Rhizogenes, development, and that's known as the hairy root disease. It looks like a root covered in hairs, but that's caused by the genes that are on this thing here. Natural system, small pieces of DNA into other cells. So this was extensively studied and manipulated so that people could take this plasmid out of the bacterium and put genes in there that they wanted. So you put what's called constructs in here, and then you culture up all these agrobacterium with the, and to your, and then select the cells that have got the antibiotic resistance and select out the transformed cells. Does that make sense? How much time do I have, Bill? I mean, I've got another hour or so.
Yeah, you got over an hour.
Oh, well, I think I'll finish in another 15 minutes or so. Finish.
Way we got you out of the room by 1 o'clock.
Okay.
Tear the tape down.
All right. And it produces plant hormones, auxin, cytokinin. Those are plant hormones, so they can influence plant growth. And then you don't need to worry about the rest of the stuff over there. TNF, it's transfer that breaks away and goes into the host cell. So this is a diagram about how to transform cells from a forest tree. And this must have been put together, obviously, because you start with but this thing here, you take a leaf off this tree, and then you do some manipulation, and you get cultured cells out of that leaf, is according to this diagram. And then those cells, those cultured cells, there's 3 different methods described here. This is biolistics. You shoot gold. You can just shoot gold into parts of the leaf. Shoot the gold particles with the gene. This is agrobacterium, where you get the plant. You get genes from in the nucleus of the cell. And this one's called electroporation, where you take naked plant cells, more or less, and you put them under an electric field, which causes DNA, those large molecules. Again, they have to be incorporated into the nucleus. This one's not very popular. This is probably the most popular method, and this is the second most popular to transform plants. When you've got the cell with the gene in, you have to select it out, culture it up, get enough. This is supposed to be the 2 roots of what you can start. One of them is developing shoots or roots to get up together, and the other one is developing from a single cell or groups of small cells, things that are somatic embryos or embryo seeds. So all of those, all the supporting in from about the mid-70s to about the early 90s. So at the end of it, what you want to get is a plant. The one plant that you get is high. And in order to get this trait into plants, you can then start breeding with this plant. And that's what they do. You've got to pick a gene that's regenerable, and it might want to commercialize. You might want conventional, so it might take a year or 2 to get the gene that's regenerated from the tissue culture system into the commercial lines of the crop. So it's a long process. Several years. So where the first plant was given to Monsanto, I think in '96, about 20 years ago. So we've had genetically engineered crops. And this information here is taken from a report that was written in published by the USDA Economic Research Service, February 2 years ago, 2014. And it says, 13,800 releases had been approved. The USDA is the body, the USDA Animal Plant Health for trial and marketing of genetic seeds and products. So this says 78,000 they'd had approved for, and was 22,000 soybeans. 78,000 releases approved for corn, 22 beans, 1,100 for cotton, and 900 for potato. Those potatoes, by the way, are Irish potatoes, not sweet potatoes. So in case you wonder.
They're not from Idaho either, right?
Pardon?
You said Irish, they're not Idaho. Idaho. This kind of potato is from Idaho.
Yeah, but you still call them Irish potatoes, don't you?
My great-grandmother's Mister, I think I've heard doing that.
You know, I was crossing Idaho one time, and the capital of the world, because you go into Idaho and a big sign says, there's a big potato sitting by the freeway on this big sign, and it says, potato capital of the world. So I thought, well, I'll stop. So I stopped potatoes, and I said, I'll have a baked potato. And they said, I'm sorry, we don't have any. And I was shocked. I said, this is Idaho and you don't have any potatoes? They said, yes, we don't have any. So, shortages in Idaho. Anyway, and what traits are there in these approvals? Herbicide tolerance is mostly Roundup. Of course, now they have some genetic resistance to 2,4-D also. With Roundup. Insect resistance. Insect resistance would be the Bt gene, the gene from Bacillus thuringiensis that and is no effect on humans. But Bt is mostly what the insect resistance is. Flavor and nutrition, 5,000 almost. Drought resistance, 5,000. Viral and fungal resistance. And this is often putting in a piece of altered virus DNA confer resistance.
Question, Doctor. You say there's no effect on humans. Why? The BT has no effect on humans. Why?
Because we don't, B protein,
Okay.
two proteins that join together.
Mm-hmm.
Insects have an enzyme in their gut that cuts the protein. It's fine. It can cleave that and break the bonds, and then there's the toxin, they die. Humans, we don't have the gene that, or the enzyme, that toxin complex.
Is it affected by pH?
Actually, we just don't have the equipment to digest it. I think I'd finish with that last one. I know. Well, the crops. Deregulation. These are materials that might be coming to the market in addition to what's already up here. But you can see corn, cotton, and then we've got tomatoes, soybeans, rapeseed, canola, potatoes, sugar beets, papaya. That's probably virus resistant, ring spot virus.
APHIS, what organization is APHIS?
Oh, this is the Agricultural Plant Health Inspection Service. It's a branch of the USDA. They're the people that rule what you can bring into the country and what you can't. You know, when the USDA inspectors, when you come back, they are actually working for these people, for APHIS, Animal Plant Health Inspection Service. But this is interesting too. Squash, rose, tobacco, flax, and chicory. I think they're chicory, kind of an odd one. I wouldn't have thought the market was big enough for chicory. Obviously, it must be.
A question, tomatoes, I see 11. Okay, a common problem is tomato splitting. Is there some GE thing that can be done to prevent the splitting of the tomato skins?
You know, I'm not sure what those are. I don't know the answer. And I think that splitting is so environmentally sensitive that, you know, the flavor saver, there used to be a tomato, flavor saver tomato. Do you remember that back from the mid 80s, early 90s? A genetically engineered tomato that wouldn't across the country, and it would still be as hard as a baseball. Well, the only problem was, yeah, to ripen. So that one didn't really go very far, but its skin was really tough. So. Yes.
So does that tell us that there's of varieties, hundreds to put into the ground?
Now these approvals up here were for.
Okay.
Okay, I didn't make that clear, did I? These are for, and these are the these that are in field trials, and you can see Monsanto, Pioneer, DuPont, Syngenta, USDA. You know, we're talking companies mostly. What used to be chemical companies. Okay.
You're right.
Okay. Any more questions on this? I may not have the answers. Okay. Now this graph by farmers, and then this, you know, this is data collected by the USDA. This is from that same publication or surveys. But the major crops that are genetically engineered, by far and away, the most important is corn. The second most important is soybeans. And the third most important is cotton. Yes, it says so right there. Probably account for almost half of the planted crop plant acreage in the United States at the moment. So half of the arable land in the United States are now just.
Does Europe come to?
No. The European Union, otherwise known as the common market,
That's why I need food in Europe is not.
they have banned they believe that the testing has not been done to show that they're totally safe for the consuming public.
So they'll wait the next
Did you
week.
bring some plants back?
Did you have any so that
Seeds.
That might be, and actually, there was a incident that was reported just over a year ago of a farmer in Washington State growing corn outside the corn, far away from genetically engineered corn. He was sending of non-genetically engineered corn to Europe, and when the Europeans are there sampling the corn, sending the samples back to their lab, one of the guys, so-called non-genetically engineered corn, they found markers that some of the corn was genetically engineered. So they stopped the whole shipment, said you can't send this to Europe. They ended up having some legal problems with because they found that the gene that was in the corn was patented. Yes, sir.
The genie is basically out of the bottom because pollen is everywhere. And
Right.
I mean, in Canada, the wheat and the canola, people have had the same issues.
Yeah.
So, to a seed now that's not some corruption would be very difficult.
It's going to get harder and harder.
Harder and harder.
Yeah, absolutely. Yes, sir.
That's just science. Opinion. So like all sorts of research and testing trials in the United States. Does the European Union have science of their own, which calls?
I think the European Union's problem is that they don't science that's being done here with the proof of to show that the quality is there. They don't think that science is being done right because most of it's being done by the companies themselves. What shall I say? Well, all of our institutions are run by humans. Humans are subjected to various pressures. Okay. You're going to do this, or let's do it this way to really be successful. Let's not worry about that right now. You know, those kind of things.
Isn't the majority of corn in the world GMO?
No. The majority of the corn in the United States is GMO. And GMO accounts for almost half GMO plant material produced in the world is produced here.
Well, hasn't it been around long enough?
Oh,
Well, we have cancer, we have viral disease, or else.
There's all kinds of things out there, but,
I mean, how would you prove if it's not in the GMO?
well, the problem is the one the first problem is this, you can't keep the pollen safe. Because the pollen goes up into the air, moves around in the wind and storms, and then gets dropped. It may all, and nobody knows how long corn pollen can be viable. If you look up and how long is corn pollen viable, everybody says, oh, 4 or 5 days. But when you start reading everything, nobody's actually tested all that.
I don't see how Monsanto is planted. And if you can't control the pollen.
Well, people, they're dealing with people at the executive level who they believe the day. I mean, Monsanto come in with cabinets full of data in his office. This says this. People won't read those cabinets full of data. It's everybody's work. Don't they? I mean,
is this for our crops? And if USDA, it is beneficial, it's not.
Say, let's answer that question in a minute.
Okay.
Okay.
What helps, comment, when I'm in Europe, I can eat their wheat, their bread, it's phenomenal. Over here, I get very allergic to it, so I'm gluten-free over here. And that's because of the GMO wheat.
But
Well, you don't know that.
wheat's not on the list.
You know, he's gonna-
No, wheat's not on the list. The seeds are not
I'll
valuable.
just tell my stomach that I can eat it.
You can eat if you're worried about GMO, because it's not GMO. Not
Maybe
yet.
other things.
It's not been approved.
Hybridize the amount of gluten
Right.
in the wheat
Yes.
kernel, so that it's not like the old original
Right.
wheat was, so that could be part of why
Right.
it is different.
You might have higher gluten wheats in the United States than other places. Okay. This just shows that what they do now is they're selling, mostly selling varieties that have at least 2 genetically engineered traits together. So this is like insect resistance and herbicide resistance. These are called stacks, and it's by far the majority of the market. Why do people want it? This is the big reason right here. Go out 3 or 4 times with different herbicides. Just go out a couple of times with Roundup. Once the leaves cover the ground, no more seedling germination. So, and of course, don't forget the sales of the seeds. Not just, and of course, I think the arrangements that engineered seeds have, or the arrangement for that, you cannot save your own seed. Corn farmers used to save their own seeds. Not anymore. Can't do it anymore. Not if you're growing GMO. And of course, this is the major thing. This was a landmark. To patent a living organism, they didn't just patent the transformed genes. They didn't say, "Oh, we put this in this organism, so we're going to patent those genes." That's got our proprietary genetic construct in it. So we're going to patent the whole thing. We're going to patent everything that's in there. All the genes in that plant now belong to the owners through the patent process. And the lines originally to incorporate genetic engineering in the 60s, 70s, and 80s public universities, like, you know, the big corn producing states, Minnesota, and Kentucky maybe even. From Georgia too. They put one gene in and said, okay, this whole cost. And the patent office.
So how many years is that for? So
I usually,
17 plus 10.
17 years,
17 plus 10,
17 plus 10.
if you
So
refile.
and you could go back to.
No, you can't.
Start
You-
growing pharmaceuticals, they come out with a generic brand for.
No, they couldn't, because Roundup resistance is already patented by Monsanto.
Well, but when will that be? Yeah.
But they,
So they both have to.
and they,
Those farmers that maybe, that pollen is a,
I think the pollen issue is going to be something that people are going to end up fighting about.
Well, because I had a question. Farm back and say.
He did, he filed a lawsuit. But Monsanto,
I thought they filed a
Monsanto
lawsuit
has a
against
lot
him
more
for
money than the
stealing.
average farm.
Yeah.
Plus they have court justices. That might make a difference too. So what about the disadvantages? Are there any disadvantages? I think this one's a big disadvantage. Contamination with pollen. And I think you're gonna we're gonna use more Roundup and more 2,4-D on all of these acres because through this genetic engineering. And because you've got herbicides and insecticides in the plants, you're going to put pressure on populations of the target pests, the weeds, the insects. 16 would have been identified in the corn growing areas now to round. This is something that, again, it might be an opinion, but some people cite lack of efficacy for consumer safety, in that it's part of a big experiment. So, to see GMO foods labeled, so and is it really appropriate to call it Frankenfood? You know, should we put bolts through the cans of corn? Just so you know. I don't. I think it's very interesting.
Dr. Garten, I have a question. Already we've already covered this. Seems to me that if the plant these chemicals, aren't the roots taking them up so that when we eat them, we organic those chemicals?
Well, the plant will be taking up the chemicals. In the case of Roundup, the enzyme that's in there breaks the Roundup down. When the Roundup gets grown, the Roundup no longer toxic.
So we're not getting the toxins.
Well, we're not getting the toxins from Roundup. And
But Doctor, you mentioned before that instead of spraying 3 or 4 times with chemicals, which may be what Europe does, okay, or in other places, we end up genetically putting that resistant so that you get a better yield and less wheat competition and everything else, right? So, I mean, you know, never is a story 100% one way or another.
Sure.
Okay, I mean, you know, you're right. Anytime a business is involved with doing something, they're trying to make a profit. We would have never got to where we are today with auto, even the food production, so without somebody making a profit along the way. So don't just discard that this, and I'm gonna use this as a test. Maybe a good thing, maybe not. Really, corn or alcohol getting into your fuel to produce more corn, more than any other crop. That pushed this genetic engineering in there because they wanted to produce a higher yield and get more alcohol in there. I mean, what's your opinion of that?
It's probably, yeah, the resurgence of corn. And I think also there is collateral advantages, because if you've got that BTG in the corn that kills the corn borer, and BTG in corn has gone up, the population. So now farmers who have not adopted genetic, that they don't have to use as much insecticide for controlling corn borer, because corn borer populations it's only an insect that's in the Lepidoptera, the butterflies that have caterpillars,
Right.
because it's the gut of the caterpillar that produces the enzyme,
So that one.
cleaves that toxin down.
Then what about bees? Is there any effect on bees? Because I know bees.
It's, the probably not influenced by, it's not influenced by BT, because bees don't go through the same metamorphic stages.
Have they made studies between Europe and the United States regarding viruses or cancers or something that would indicate that it's bad here for?
Not that I've seen,
Not yet.
not yet, maybe in the future.
So what about organic?
So, right, there's a question over there. Yes.
Dr. Garten, Monsanto and others equivalence justify or to suggest that GMO modified foods are substantially to non-GMO modified foods. And since they have accepted this quote unquote gospel, and others have as well, do you think substantial equivalence be 100% justified based on what you know about scientific evidence?
I would say that, to my mind, as an individual, I think it's a stretch. Because genes in there, and those genes, even though they're, the plant produces own gametes for sexual reproduction, makes its pollen and its egg cells, it goes through a meiotic process. And during the meiosis, chromosomes can switch around. Pieces of chromosomes from the analogous chromosomes, homologous chromosomes come together. So that means that the parts of the construct are addressed. I think that there's a lot of unknowns. And for Monsanto, Monsanto is a big company. They were so they're in their minds it's safe, but it may not be safe for everyone. But I think each person has to make their own decision with their understanding. But I don't think, I think the doctor actually equivalency. It sounds nice, but I think when you think about it, it doesn't stand up. At least to me.
What about beans? Beans, because a large percentage of the corn that we grow is fodder for meat and poultry, right? So, and high fructose corn syrup, you get all these one-offs.
Well, I think animals have also been subject to genetic engineering. We're talking about plants. But you have got the ability to move protein enzymes from one organism to another. So you can get